So humans are the most evolved mammals and most evolved animals in the world. And as we evolved, we lost a few really important features and gained some other ones. And as you saw from Dr. Bainey's introduction, certain animals retain the ability to completely regenerate limbs, like the salamander or newt or axolotl. And unfortunately, most mammals, and certainly humans, have completely lost the ability to regenerate even small parts of their limbs, with the exception of just the tip of your finger. There are a few tissues in the body that still can regenerate, like bone, muscle, liver. It's more of a repair, but they're able to heal. And a bone, after it heals completely, will have a very hard time finding where that injury actually occurred. But the tissue that we're interested in, which is articular cartilage, lines the surfaces of all of your movable joints. It's this white tissue, and this is a human knee where you can see the cartilage, has really no capacity for repair. And you can see if you get a damage or injury to your cartilage, it never repairs. And this focal defect here eventually progresses to arthritis, a really painful and debilitating condition where the only real treatment is to replace that joint with a metal and plastic prosthesis, as you saw in Dr. Kosker Fernandez's talk. Now, if you're 75 or 80 years old, this is a great surgery, and it'll replace your limb, and you'll get mobility back. But the problem is joints like this. And this is a patient. You can see this patient has already had a hip replacement, and the other hip looks okay on an X-ray, but our surgeon, Dr. Clohisi, went and looked in there, and you can see the cartilage is completely broken off and damaged, and this patient needs a second hip replacement. The problem is that this patient is 22 years old and got this first one as a child, and this is not a great surgery for a 18 or 20-year-old because these replacements only last 10 to 15 years, and then they have to be replaced again and again. What we need is not to replace that joint, but to try to regenerate it. And there's a tremendous need for tissue regeneration. If we look at all the different types of organ failure in the body, they add up to an enormous cost to our economy of close to $1 trillion a year. And this includes joint replacements, skin failures from burns and diabetes, heart surgeries, organ transplants, where if you look at our waiting list for organ replacements, they continue to increase every year, whereas the number of donors available for lungs, kidneys, and hearts doesn't increase. So with all of these, we need the ability to regenerate and not replace tissues. And when I started in this career, this article came out in Life magazine about how to bioengineer a new body and how to build a new body. And you can see it's almost all replacements, metal and plastic, and using biomaterials and materials at that point to create replacements such as a joint replacement, a prosthetic limb, an artificial heart. 10 years later, Time magazine came out with the next version of this, which is, can we regenerate our body? And with the field of tissue engineering, can we grow replacement parts to try to regenerate instead of replace with metal and plastic? And so, as you saw earlier, there's several different ways to do this, but the main component to regeneration is living cells. And how do we harness living cells to rebuild tissues and organs? There's a few different ways. One is to take stem cells or other cells and inject them into the body. This is called a cell therapy. The other is tissue engineering, as you just saw, where we combine cells, materials, and molecules or environments to regenerate tissues outside the body or even inside the body. And a third way is gene therapy, where we can correct genes or modify cells and insert new genes so they can provide a therapy for the body. But the first question is, if cells are the main component of these therapies, what is a cell? And we define cells really as the fundamental unit of life, and we define it by the presence of the genetic code, that cells have DNA that tells them what to do. And there are all different kinds of cells in the body. They all have the same code in your body, but the way that the DNA is wrapped up and exposed makes them act like different entities, and they have different properties as different cells. And so the way nature builds a joint is from embryonic stem cells that differentiate, and to get a joint, you need all different types of cells. So you need bone, cartilage, tendon, ligament, fat, blood vessels, and so on. All of these come from one cell originally, which expands, turns into progenitors to other types of cells, and creates these in the body. So what is a stem cell then? You probably hear a lot about stem cells in the news and on billboards and in your in-flight magazine. It actually has a very simple definition. It's a cell that hasn't decided what it wants to be yet. So it's kind of like a teenager of a cell that it's growing up. You may want it to get a job and do something. You probably don't want it to divide, but you have to tell it what to do. And the simple definition is a cell that can replicate itself and form more stem cells, or it can differentiate into other cell types. Now, you've probably heard about stem cells quite a bit, but they're everywhere in your body, and they're necessary for your body to survive. And we create millions of new cells every second in our body. So for example, your bone marrow produces blood cells at the rate of about 2 million a second. And if you didn't do that, you would run out of blood cells within a few weeks. Without stem cells in your body, you would lose your gut in about 2 days because it turns over so rapidly in that harsh environment. Your skin turns over every 3 weeks, and as I mentioned, your blood every few months. And this is because it needs to propagate. The stem cells form a new stem cell and a differentiated cell. So where do we have large amounts of stem cells in our body that we can use to create new tissues? Well, Dr. Grayson mentioned it. It's in our subcutaneous fat. There's also cells in our bone marrow, other tissues, but as a source of stem cells where we can take some very readily using liposuction or other techniques, subcutaneous fat is an extremely rich source of cells. And we discovered this many years ago working with Dr. Jeff Gimbel, that we found that cells in human subcutaneous fat that we got from liposuction waste could be converted into all different cell types. So from just one cell source, we could make cartilage, bone, fat, even nerves and muscle. Now, how do we use these stem cells? So this is a typical ad you'll see in your in-flight magazine, and it'll tell you that stem cells are specialized cells that know what you need, where you need it, and they go there and they become it. And that's completely not true. The unfortunate thing is that these types of ads are everywhere, and there are now over 600 unapproved stem cell clinics around the United States and many more overseas for medical tourism. And they're a cause of a lot of concern because they're not regulated. Most of them are not actually using stem cells as a therapy. They're injecting what we call technically gunk into your body, and it goes away very fast, and they charge thousands of dollars. And there's a bit of a placebo effect, so there's anecdotal evidence that it works, but there are no randomized clinical trials that show that these therapies actually work. So we don't recommend any of them. The approach that we use has been tissue engineering, as Dr. Grayson showed you, where we take stem cells and we actually reprogram them and we change their DNA so they do what we want them to do. We put them on a biomaterial scaffold to grow cartilage, which is in the shape of an entire joint. So we can actually try to replace that joint surface with living tissue instead of with metal and plastic. And you saw some information on different scaffolds. They're all different types that have been used for cartilage, things such as gels, which are like the properties of Jell-O. The cells love to grow in them, but they're not very strong. Fibrous materials, think of a cotton ball as a fibrous material. Again, cells can grow well in it, but it doesn't have great properties. And 3D printed materials, which, as you saw from the last two talks, are fantastic for making bone structures. But for cartilage, we need something that's flexible and very tough and very strong in one direction, but softer in the other direction. So what we developed was a way to make a fabric that has the properties of cartilage. And we developed a technique called three-dimensional weaving. So unlike 3D printing, where we put down one layer at a time, 3D weaving takes hundreds of fibers, and these are materials that the body can resorb slowly over time, and weaves these 500 to 600 fibers into a thin layer, which is the thickness of cartilage. And you can see the structure of the material, and it's porous, as Dr. Grayson described that you need it to be, and we can tune the properties. We can weave it tightly or loosely, and then we can fill it with cells so it grows a tissue replacement. The nice thing is that it's three-dimensional, so we can actually make this in the exact shape of a human joint. And here, if we take a MRI or CT scan, we can make a mold in the shape of the joint, create a scaffold out of this resorbable biomaterial, grow cells on it for 1 month, and we have an entire surface of a hip that we can use to replace the cartilage. And so the idea is that if you have osteoarthritis, you're too young for a joint replacement, we can take some stem cells from your bone marrow or from your subcutaneous fat, grow them in the shape of a hip, and just resurface the parts that have worn out so that you can avoid that joint replacement. Now, the problem is this goes back into the joint, and the joint in arthritis has something called inflammation, where the cells make enzymes where if this is normal cartilage, the red stain shows newly formed cartilage. In the presence of inflammation, the cells break down that cartilage and digest it away, so you have the same problem again. So we thought, what if we could make a cell that could fight that inflammation and make cartilage at the same time? So if we think of a cell, it's a little object like this. It has a nucleus, organelles, parts that let it do what it wants to do. My background is engineering, so I think of a cell more like this, where it has microchips, hard drives, motors, and computers. And what we really need is a combination of the two, where we can engineer the biology of the cell and create new cells that can do what we want them to do. And so what we did was try to create cells in our lab that are smart. So a normal cell that sees inflammation has this damage response and digests the tissues around it. It's done in a manner that the cell is trying to fight off bacteria and other invading pathogens, whereas in a situation like this, it'll just break down your newly formed tissue. What we wanted to do was get rid of that damage response in the cell, and what we did was use a new developed technique called CRISPR, which is a bacterial system that lets you cut and paste pieces of DNA. And what we did was rewire that cell so that instead of having a damage response, it actually made drugs that fight inflammation. So now whenever this cell sees an inflammatory response or an inflammation in the joint, it makes drugs that fight that inflammation and block it. So the cell is self-regulating, and then it turns off the drug delivery system whenever the inflammation has gone down. And what we showed is that where inflammation breaks down the cartilage, these self-regulating cells can protect it. So the overall concept now is if we have a cell, we can use what's called gene editing or CRISPR-Cas9 gene editing to rewire that cell and put in logic circuits that we really want the cell to have that don't occur naturally. So these are cells that don't exist in nature under normal conditions. And then we put them on that scaffold, and the scaffold is in the shape of a joint. The cells attach to the scaffold. We then treat them with growth factors so that they make cartilage-producing, turn them into cartilage-producing cells, so that now they synthesize all of the parts of cartilage, and then they replace that scaffold with cartilage, and then we hope to implant this in the body. So as I mentioned, 1989, the future was metal and plastic materials. 1999 was regenerating tissues. And we think the future now is the post-genomic area where we combine all of these technologies, where we can create new materials, new tissues, and even new genes and new cells to replace our body parts. And with that, many thanks to the funding organizations that support our work, particularly the Shriners Hospitals, the NIH, the Arthritis Foundation, and of course, my fantastic lab who did all of the work in this presentation. Thank you.